In this video, you should understand the general overview by showing the step in gene cloning. You should also be able to describe the step in gene cloning by using plasmid as the vector. Gene cloning is the process to produce genetically identical copies of a certain desired gene. It is usually involve plasmid as a vector and bacteria as a host cell. Gene cloning is broken down into 5 distinct steps. First is isolation, where a plasmid is isolated from bacteria and gene of interest from the source DNA. Second is the cleaving or cutting of the DNA, source of DNA and also plasmid DNA. Both use the same restriction enzyme. Third is the insertion. The gene of interest is inserted into a plasmid by using DNA ligase. Fourth is transformation and amplification. The bacterial cell is transformed when the recombinant plasmid is reintroduced into bacteria and the transformed bacteria is undergo amplification process. Lastly is screening. The transformed bacteria colony must undergo screening to identify the bacteria colony that contain the DNA recombinant.
So, now let's look at the step more detailed. So, we look at the step is more detailed. In isolation, both the donor DNA, target gene, gene of interest and plasmid are isolated. Gene of interest is isolated from DNA source, which plasmid is isolated from E. coli. The plasmid and the gene of interest are cut by the same restriction enzyme to produce complement sticky end, and the cutting region is known as a restricted site and it contains the same palindromic base sequence. This step produces thousands of DNA fragments and DNA plasmids. As seen in figure, the plasmid is cut only once to create a single opening, while in the donor of DNA is cut twice to extract the small section of gene of interest from the donor DNA strand. Insertion occurs after mixing between the plasmid and target gene, where the sticky end of gene of interest cut the plasmid from the complement pairs. The complementary sticky end is ligate by DNA ligase, thus producing recombinant DNA, also known as a recombinant plasmid. The recombinant DNA is transformed into the host cell, such as E. coli, by electrical shock. Then amplification process occurs to the colony of bacteria, producing 3 types of bacteria colony. The first type, bacteria has recombinant plasmid with a target gene. Second, bacteria with no plasmid. And the third, bacteria has self-ligate plasmid, meaning it does not contain gene of interest.
So now we need to identify the bacteria that contain the recombinant plasmid. So a screening process is required. So E. coli colonies are cultured on a medium with antibiotic ampicillin. It is used to detect the bacteria containing recombinant plasmid and eliminates the bacteria without the plasmid. This is because the transformed bacteria with recombinant plasmid contain ampicillin-resistant gene. So, only bacteria containing plasmid that have antibiotic-resistant gene can survive and grow in the medium. So, as shown in the diagram here, okay, can you see it? So, as I said before, bacteria with plasmid will only survive after 15 hours, 15 or 16 hours, while bacteria without plasmid will die or be eliminated. So after that, the survival bacteria colonies undergo further screening, which is called blue-white screening. The survival bacteria colonies is cultured on medium with agar, which is white in color. The agar is the substance similar to lactose that helps to identify the plasmid that carries the target gene. The bacterial colonies without the target gene will turn blue in color. So remember, without target gene, turn blue in color. So this is because the LAC-Z gene is still functioning. So if the LAC-Z gene is still functioning, means it can produce beta-galactosidase, which hydrolyzes the X-cal from white color to blue color. So that is why the colonies or the bacteria without the target gene will turn blue in color, while the bacterial colony with the target gene remain white in color. Here, the LAC-Z gene becoming non-functional or not functioning. This is due to the presence of target gene inserted in the LAC-Z gene region. So when the target gene is inserted in that region, so the bacterial colony lack the beta-galactosidase, meaning it will not produce the beta-galactosidase. So the white colonies show bacterial clones which carry recombinant DNA clones. So as shown in the diagram here, so as you can see how blue-white screening works. The plasmid without insertion with gene of interest will turn blue in color. So without blue, okay, remember. So blue due to the LAC-Z is still functioning, okay. So without blue functioning, so that is the keyword. So while the plasmid with insertion, okay, of the gene of interest in the LAC-Z region maintains white in color. So why? Because the LAC-Z is disrupted or not functioning.
Okay, first step is isolation of gene of interest from donor DNA and plasmid from bacteria. Second, donor DNA is cut twice while the plasmid is cut once by the same restriction enzyme. Third, gene of interest is inserted into the plasmid and then ligated by DNA ligase producing recombinant plasmid. Fourth, the transformation process occurs when the recombinant plasmid is introduced back into bacteria. Then, the bacteria is replicated through amplification process, forming bacterial colonies. At the end, the bacterial colonies must undergo screening process to identify the desired bacterial colonies with the gene of interest by ampicillin screening and blue-white screening.